Greetings all. My name is James Pelletier. I've just started as a junior group leader at the National Biotechnology Center in Madrid, and I'm delighted for this opportunity to continue the third day of the Minimal Cell Workshop. And following Karim's really inspiring talk, this talk too will be about how cell division is not a process that happens in isolation in the genomically minimal cell JCVI-syn3B, but rather reflects the integrated physiology in a number of physiological processes inside of the cells. I'll be presenting on behalf of this collaboration between a team at the J. Craig Venter Institute led by John Glass, with major contributions from Kim Wise and Li Sun, a team at the National Institute of Standards and Technology in the US led by Elizabeth Strakowski, as well as co-authors at the MIT Center for Bits and Atoms, where I was a graduate student, as well as UC San Diego. A question motivating this work was, what genes does the genomically minimal cell JCVI-syn3A, syn3B require for normal cell division? JCVI-syn 3.0 was chosen for its ability to form colonies, its ability for populations to grow. But when we performed microscopy, the cells exhibited extreme variation in cellular size and shape, with some cells several hundred nanometers in diameter, similar to wild-type JCVI-syn 1.0, but some cells several microns in diameter. And a 10-fold difference in diameter corresponds to a 1,000-fold difference in volume. So these are drastic changes in cellular size. The identification of JCVI-syn3A largely restored the normal cell division exhibited by wild types in 1.0, and JCVI retains an additional 19 genes. So a question was, which of these 19 genes are needed for the cells to divide? And the genes were organized in 8 clusters of adjacent genes that likely represent transcriptional units, as well as a second copy of the ribosomal RNA operon that wasn't needed for normal cell division. And the first cluster that caught our eye was this cluster that includes the bacterial tubulin homolog FTSC, as in Karim's last slide. This forms filaments that often localize at the division site. It's highly conserved in bacteria, and it participates in cell division in most bacterial species. In many bacterial species that have a cell wall, it coordinates peptidoglycan synthesis. Of course, the mycoplasmas don't have a peptidoglycan cell wall, but it's still involved, it's still retained in mycoplasmas, and it participates in cell division. And here I just want to note that an advantage of using the minimal cell to study cell division is not only its genomic simplicity, but also its mechanical simplicity, because without a peptidoglycan cell wall, which is a kind of elastic plastic structure, here we think, but haven't experimentally proven, that the membrane behaves more as a fluid, and that simplifies the mechanical description of the cell division process. So upon observation of this cluster, we thought, oh, if we restore this cluster to the minimal cell syn 3.0, that will restore normal cell division. But that didn't happen. The cells still exhibited extreme variation in cellular size and shape. And looking at what was known about gene function, there were no other hints. So Li Sun, who's the co-first author of this work, undertook a systematic study, and these 7 genes, 5 of which did not have a known molecular function, were all together required to restore normal cell division in the genomic context of syn 3.0. So this perhaps isn't, it's a hint that these genes could be doing something, but we really don't know molecularly how are they contributing to cell division. And FTSZ alone is not sufficient, but in this context it was needed. Perhaps more surprisingly, if you start with JCVI-syn3A and you knock out just that FTSZ containing cluster, the cells still look largely normal. So this could be an interesting strain to study for the microcalorimetry experiments. And it also suggests that we need to think about this division process beyond this cytokinetic Z ring, but what other forces could be contributing to cell division in these genomically minimal cells. So that leads to this work, which was led by David Bianchi in the group of Zanluthi Schulten. Although this work is a proteome-wide analysis, so for anybody interested in other aspects of the proteome of JCVI-syn3A, this work is a proteome-wide bioinformatic analysis, including all of the AlphaFold 2 predicted structures. Here I'll just focus on the genes that were needed for cell division. So this is now our current knowledge of genes that could be contributing to cell division in syn3A. The blue genes are the 7 genes that we identified. The gray genes are genes that are retained in syn 3.0 as well. And the first two, EZRA and GPSB, we hadn't annotated them previously. In David's work, identified these homologs, and these are known to interact with the division machinery in other bacterial species. But at a systems level, we still don't understand how these genes, as well as other genes, are contributing to the process of cell division. And as the mechanisms remain unclear, we're currently pursuing a combination of biochemical reconstitution and biophysical modeling to guide hypotheses as to how these genes could contribute to cell division. So we've just started a collaboration between the groups of Germán Rivas, Silvia Zorrilla, and Begonia Monteroso at the Center for Biological Investigation in Madrid, as well as the Chemical Physics Institute in Madrid, as well as Saul Ares at the National Biotechnology Center and John Glass at the JCVI. And Germán, Silvia, and Begonia are world leaders in purification and biochemical reconstitution of bacterial division proteins such as FTSC and proteins that interact with FTSC. And they've worked extensively with those homologs in E. coli and other bacterial species, and have reconstituted interactions between those proteins in various biochemical formats. So the goal of this project is to perform similar experiments, but with the proteins encoded by the Mycoplasma mycoides syn3A genome. There are interesting sequence differences between these homologs. For example, this is the C terminus of FTSE shown. In FTSE, the C terminus is known to affect lateral interactions, as well as interactions with other proteins that anchor it to the membrane. And in contrast to the E. coli and Bacillus subtilis versions of this protein, the Mycoplasma mycoides has a ton of negative charge in the C terminus. And so we expect there will be interesting biochemical differences between these purified proteins, and we've just started this work. Regarding the biophysical modeling, I'll describe an opinion that I wrote with John Glass and Elizabeth Strakowski. So here, when we grew cells in microfluidic chemostats, and then I took picture every half second to observe the shape fluctuations of the cells, we observed that they fluctuate on the sub-second time scale. And we haven't quantified this yet, but this is at least so far, it's qualitatively consistent with having a fluid membrane, which is also consistent with not having a peptidoglycan cell wall. And there's a language of membrane physics that can predict the shapes of vesicles that have a fluid membrane. So this is theoretical work that has a history of decades, with major contributors including Wolfgang Helfrich, Ouyang Zhongkan, Reinhard Lipowski, Udo Seifert, Sasa Svetina, and others. The key point here is this is a phase diagram that has 2 inputs. So for any pair of inputs, the model will predict a shape that minimizes the bending energy of the membrane. The 2 inputs are the surface area to volume ratio on the horizontal axis, where 1 is a fully inflated sphere, and as you move to the left, the sphere deflates, so it could take on more shapes. And the vertical axis represents the preferred curvature of the membrane. And when I first became aware of this model, I thought this is so cool because there are parts of this phase diagram that represent spontaneously constricted and even fully constricted vesicles that look like a dividing cell, even though the vesicles don't have dedicated cell division machinery. This is just the overall membrane curvature driving this. And Sasa Svetina has described shape trajectories in this phase diagram that look like cell division cycles. So we don't wish to claim that this simplified model is the model that describes the process of cell division in these bacteria. However, we think it definitely provides a biophysical context in which dedicated specific protein and lipid protein interactions could modulate cell division. And we're also taking this model seriously and quantitatively. So this is work from, again, from Sasasvatina, that has considered the shape trajectories of growing vesicles, both the volume and the area, and how those increase over time. And we can imagine a shape trajectory of a dividing cell of syn3A perhaps looking something like this, which would look like this green line on the phase diagram. So this could perhaps represent the shape of a syn 1.0 cell over the cell cycle. But syn 3.0 perhaps undershoots this line, which is a critical line at which the cell divides, and enters this really complex region of the phase space where the shapes are very complicated. So in other words, maybe Syn 3.0 just doesn't generate as much membrane curvature as Syn 1.0, and there's many ways in which a cell could generate membrane curvature. Furthermore, from this work from Sasas Vatina with a master's student through Gaitz Ortiz, there's an expression in this paper that the size of the vesicle should be a function of the doubling time, the growth related to the growth rate, the membrane permeability to water, and the bending rigidity of the membrane. We know the doubling time from experiments. We haven't yet. James Sayens has done experiments estimating the membrane permeability to fluorescein diacetate, and I'm curious how that relates to the membrane permeability to water. And this value of the bending rigidity is very common for biological membranes. And if we plug these values in, we get a radius which is comparable to the actual cell size of JCVI-syn3A. So we're interested currently in testing these quantitative models and relating them to the cell division process. So the focus of my group, which has just started in July, is using biophysics, mechanics, and thermodynamics to help relate the molecular composition of the cell to the physiology of the cell, currently with a focus on cell division. And with that, I'd like to thank all those who contributed to the work and the members of my group who just started, Paula Perez and Alejandro Serrano, who will be starting this month, as well as collaborators that I've already described, and other collaborators, and really thankful to be a part of this community and looking forward to your feedback.
James, I'm psyched. I can't wait to be more a part of this and to hear to see where this goes over the next few years.
Thanks,
how much, so what are the key technologies that you anticipate bringing to this work that we haven't seen in our field yet?
So I think that a major challenge, so I think in order to test this model experimentally, so if I go back to this phase diagram, an ideal experiment would be to image single cells over time and perturb them in both directions. So we could do osmotic shifts to take spherical cells and deflate them by putting them in hypertonic solutions, and then returning them to hypotonic solutions to move along the horizontal axis, and then along the vertical axis, ways to perturb membrane curvature. We hypothesized that crowding of lipoproteins might be a dominant contributor to the generation of membrane curvature. So being able to control the density of proteins on the surface would be really helpful. And then also, for example, James Dobbin's spec showed cleavage of lipoproteins with phospholipase D. So one hypothesis would be if we were to cleave those lipoproteins, that would potentially reduce curvature, and then cells would move down away from these more curved shapes to more prolate ellipsoid shapes. Now, the technical challenge is, how do we measure the shape change of a single cell over time without perturbing the cell? Because in our previous experiments growing cells in microfluidic chemostats, we observed that if we image the cells too often, that does have a detrimental effect on the growth rate. So we're interested in starting a collaboration with a group of, with actually a number of collaborators at the Universidad Autónoma de Madrid, primarily the group of Pedro José de Pablo Gómez. And they're doing atomic force microscopy of large viruses. And AFM could provide a way to image shapes with the required resolution without having phototoxicity. But that's not entirely new technique for the community, because I know that Daniela Bittencourt has described work with AFM, and we've been discussing with Daniela complementary approaches. And I know that Makoto Miyata's group has used AFM extensively for other studies with mycoplasmas, but I think AFM may provide that sweet spot between enabling dynamics without poisoning the cells from illumination. So But even if we can't do dynamics of a single cell over time, I think the electron microscopy techniques that that other groups are working on will be really helpful, because we could do population look at the shapes in populations and then perturb the population and then look at the shapes and then try to relate them. So it's an exciting combination of approaches.
Can you speak for a second about, so we're looking at this genetically extremely simple organism. What you observe with this, how can you relate your findings here to cells in general, as opposed to our esoteric niche, an organism we love dearly? But how does this, in your mind, relate to the rest of cell biology?
I think the two most closely related biological systems that I find really inspiring are, on the one hand, L-form bacteria. Those are many bacterial species that have a peptidoglycan cell wall. In some cases, under stress, will no longer will lose the peptidoglycan cell wall, but they remain viable and they grow in the absence of the cell wall. And these cells often exhibit extreme variation in cellular size and shape, similar to SYN3.0. And I've never seen an L-form situation where the cells exhibit, say, symmetrical binary fission, similar to many mycoplasmas that don't have a cell wall. So it'd be really interesting to try to restore something that looks like symmetrical cell division in an L-form bacteria. And then the other bacteria that I find fascinating are the obligate intracellular parasites. So these are bacteria that have even smaller genomes than SYN3.0, but they're not free living. They need to live inside of another cell. So those bacteria are also often pleomorphic, and the mechanisms of cell division in those bacteria is also pretty wild. So those are the two closely related species. I think thinking about the inner membrane in bacteria that has a cell wall, say gram-negative bacteria, is interesting, but as there's extensive coupling, mechanical coupling between the inner membrane and the peptidoglycan cell wall, I think it would be hard to map these insights onto that system. But general processes involved in surface area to volume regulation and mechanisms that cells can generate curvature may map to some extent, but with further modification.
James, this is Bert Polman.
Oh, hi, Bert.
Yeah, I missed your first few minutes, but very nice. Thank you very much.
Thank you.
Could you go back a few slides where you showed the N-terminus of one of these FTS proteins? I think it was FTS.
It's the C-terminus.
Yeah. So where you see the charged tail. So two questions. You say that FTSC is not essential in mycoplasma.
This is correct.
Did you ever look if the FTSZ ring is formed when you have it present? And the other question is related to this sequence that is highly charged. So to me, it looks natively disordered, and well, nowadays natively disordered regions also tend to form condensates. So is there a link that perhaps this FTSZ protein, well, perhaps is doing something differently and is forming some sort of a condensate here in JCVI?
Thank you very much, Bert. So to your first question regarding the spatiotemporal organization of FTSZ in the strains that do have it, I have not imaged it, but the Ji-Wong Kim and the group of Taekip Ha has ongoing work looking at fluorescent imaging of FTSZ. And thus far, I think there are some examples where there seems to be more fluorescence at the area where the cell is dividing. But I think that's an ongoing study. And I think that, the dynamic process of oligomers forming and how they become enriched at the division site, as has been extensively studied in E. coli. I'm remembering a paper from the University of Tennessee, I think, but I don't remember the name of the well, it will come to me later and I'll drop it into the chat. But I'm aware of some really beautiful super resolution imaging of the dynamics of FTSE formation in other bacteria. I think a technical limitation here remains the phototoxicity. So we'll have to overcome that. But anyways, even looking at fixed cells, there is some hints that FTSE is enriched at the division site, but quantitatively what fraction of its partitioning there and other aspects of its organization, I think, remain to be studied. Regarding formation of condensates, our collaborator Herman Rivas, for example, has studied condensation of FTSE in the context of interacting with another protein called SLMA, which is involved in a mechanism called nucleoid occlusion, where bacteria tend not to divide in the places where the chromosomes are. And so this helps localize division to the midplane after chromosomes have segregated. However, we're not aware of any nucleoid occlusion factors that are retained in the genomically minimal cell, although it's possible they haven't yet been annotated. So, but anyways, I agree that that phase separation and condensation of FTSE could be at play here, and I'm curious. I think that in other species as well, the C terminus is also described as disordered. And then the C-terminal tail, CTT, and the C-terminal variable region, CTV, have differential effects on lateral protofilament interactions with other, for example, SEPF and FTSA. And another interesting feature of the JCVI-syn3A protein is that it entirely lacks this C-terminal variable region. So, but no, I agree. Both are interesting directions. So thank you for the feedback.
Thank you, James.
Okay, so moving on, it is with great pride that I turn it over to our next speaker, Sage Glass, who's going to talk about our work to understand how the addition of the non-essential pyruvate dehydrogenase genes back to the minimal cell alters metabolism. Sage, the floor is yours. Sage, are you with us? This is not a good sign. There we go. You're on. Although we don't hear anything yet. Are you muted? Sage, we cannot hear you. Sage, we can't hear you. Okay. We're getting there. We're working on it. Patience, everyone. So she left, and I see her outside my office. There are people there trying to help her. Tell you what, we're going to pause on Sage. Manuel, would you feel okay to going out of order, please? Maybe not. Luckily, we're all very patient. Kareem and James, while we wait for Sage, the systems that you talked about, if we could synchronize cells, and this is something that James Pelletier has been talking about, how would that possibly alter the way you approach things? Well, we can talk about this offline.
Well,
We're
short
getting
answer,
closer.
if we can synchronize cells, that would be awesome. So, yeah.
Yeah, I think you guys can.
This is something that James Pelletier and I have been talking about, and is part of the project that we have going on. But this is, we do believe it is doable. Maybe
Cool.
in sort of a Caulobacter approach, that you fix cells to a surface and then collect daughter cells at
yeah.
specific intervals.
Yeah, I would be very interested to get involved with that.
Related to that, what approximately how many cells do you need to generate what was it, a microwatt? Do you have a sense of?
Yeah, we typically start. We typically start with some 500,000 to 1 million of cells, and then let them grow. And in total, they double kind of 5 times in our machine. So 2 to the 5 is the factor by which they increase.
Right. Thank you.
Would the slightly faster growing cells that have pyruvate dehydrogenase, which we're about to hear about, would that help what you do or hinder it?
No, it's neither helping nor impeding anything. We only have to make sure that they don't grow too fast. That was something that surprised us as well. So still the syn3B cells grow very fast. So our initial OD is 0.0005 OD 600. It's a very small amount that we add. Because we need sufficient time for them to grow up, but we can have them run for 2 weeks or 1 day. There's no restriction.
Okay.